Good morning, everyone. I'm Amrita Ahluwalia, the Secretary General of this Congress, WCP 2023. I'm also Professor of Vascular Pharmacology at Queen Mary, University of London. So it is my great pleasure to be introducing this morning's keynote speaker, Professor Dr. Joan Brown. Professor Brown is Distinguished Professor and past Chair of the Department of Pharmacology at UCSD, University of California, San Diego. I think you all know her for her amazing work helping us to understand the key role of GPCRs in cell hypertrophy, proliferation, and inflammation, particularly within the heart. And in fact, I know Professor Brown's work very well because I'm very interested in inflammation and cardiovascular disease, and actually the work that she's done really has cemented and developed our understanding of the key role of inflammation in driving cardiac dysfunction and ultimately heart failure. Professor Brown has won many prizes and awards, not least the AHA Basic Cardiovascular Sciences Distinguished Achievement Award. So it is my enormous pleasure to invite Professor Brown to give her plenary lecture. And in fact, I feel a bit like Alice, waiting to be taken through the looking glass by Professor Brown to hear about G proteins and their lessons for cell growth, survival, and invasion.
Well, thank you so much for that kind introduction, and I am truly, truly honored to be here. Just a bit of a correction to my title and talk. I had to give a talk last night for someone who I work with who couldn't come, and that was in the sort of inflammation, cardiac inflammation area. So my title no longer has inflammation, it now has invasion, but generally I will give a pretty broad talk. And for those of you who wonder what the title means, not everybody knows, it comes from a book by Lewis Carroll, who is a Victorian mathematician turned children's author. And this is the sequel to his book Alice in Wonderland. And as it turns out, a looking glass, which I had to look up to make sure I was right, is actually a mirror. It's not a binocular type of thing, it's a mirror. And so because it's a mirror, that gives me license to sort of do reflections. And I'm going to reflect on something, you'll see what I'll reflect on, but I'm going to reflect on things with a focus on what one of the themes of Lewis Carroll's books, and this one in particular, are, which is how what seemed impossible came to make some kind of logic in the end. And I'm going to go through basically this list of GPCRs, all of which I've worked on in some sort of chronological order, to kind of explain how I got from point A to point D, which is where I'm going to end my talk. And for those of you who work on GPCRs, my apologies. This is going to be very foundational. For those who don't, hopefully it will be a good introduction for the many symposia that follow in this area. So to kind of begin at the beginning, when I began graduate school, and I know this dates me, but some of you know how old I am anyhow, this paper was published, and it was published by Earl Sutherland, who went on to win a Nobel Prize for this work. And the paper says that adenyl cyclase is basically the or an adrenergic receptor. And that was a great theory, a good idea, and it was an idea because Sutherland and his collaborators had shown that norepinephrine and epinephrine, adrenergic agonists, in increase the formation of this new second messenger they were measuring, cyclic AMP. And so the model, the sort of logical model in their minds and in the minds of most, was a model that looked something like this, where, let's see, by that pointer, where adenyl cyclase had a catalytic unit and a regulatory, which is what R is here, unit, which they surmised was the adrenergic receptor part of the adenylate cyclase molecule. So I went into this thinking that this was the case, and also I came to graduate school having been a neurobiology major, and so I was interested in looking at this system in the brain. And I don't know how many of you know of or have tried to use a McElwain chopper to make brain slices. Anybody in this room? Martin, yes, Paul. So it was nearly the end of my graduate career. There's no way you can make slices of brain with this thing. Unfortunately, my thesis advisor had the idea that we could instead use the, let's see how this pointer works here. We could instead use the retina, which is like a little piece of central nervous system at the back of the eye. It's very easy to take out. It's very easy to oxygenate in an organ bath. And so we used retinas. We actually got them from a slaughterhouse. They were bovine. They came every week. It was very handy. And I did the experiment. Okay, let me look for adrenergic stimulation of cyclic AMP formation in this retina. And much to my surprise, I found that dopamine, I'm not sure why I actually tested dopamine along with norepinephrine and epinephrine, worked very well, and norepinephrine and epinephrine didn't elevate cyclic AMP. And furthermore, this cyclic AMP formation was not blocked by propranolol, phentolamine, by adrenergic blockers. It was blocked actually, not in this paper, but in a subsequent one, by phenothiazines, chlorpromazine, drugs like that turned out to be antipsychotic drugs, and we now know that the dopamine receptor is where the antipsychotic drugs work. Another thing I just want to point out, very small here, is this was communicated by Alfred Gilman, and this is not the Al Gilman you all know, but the elderly father of Al Gilman, who was the chair of my department at the time. So dopamine stimulating cyclase, that wasn't supposed to be the case, and so that's my first, how is this possible? What is the logic? And sort of probably 2/3 of you in the audience, of course, know already that the reason this is possible is because it's not one enzyme, that's the adrenergic receptor in cyclase. In fact, there's a G protein, and the first one that was discovered was GS, and actually was discovered by Al Gilman Sr.'s son, Al Gilman Jr., and so this G protein, GS, couples to many receptors, the dopamine receptor, the adrenergic receptor, and multiple peptide receptors, all of which stimulate adenylate cyclase and cyclic AMP formation. So, okay, that actually turned out to make some sense. It turned out to be fine. After my postdoc, and that work was not on G protein, so I won't talk about it, I went to UC San Diego. And at UC San Diego, I used actually another model system, kind of similar to the retina, and it was the murine atria. It's actually the atrial appendage, very handy to kind of like snip off the top of the heart, also something you could put in and oxygenate in an organ bath. And using this system, I looked at the effects of acetylcholine. I actually used carbachol, stable analog, thinking it might also stimulate cyclic AMP formation, but in fact it didn't. It inhibited cyclic AMP formation, particularly if you use an agonist like isoproterenol, you could see very marked inhibition. And believe it or not, that was pretty novel at the time. I remember presenting at an experimental biology meeting that there was this cholinergic antagonism of the adrenergic cyclic AMP response. And again, one could say, that doesn't make sense. Why is it not stimulating adenylate cyclase? And that's because we didn't know it until shortly thereafter that there was actually another G protein, another pathway, and that was the G protein that we now know of as being GI. And the muscarinic receptor acts through GI to actually directly inhibit the activity of adenylyl cyclase. And there's a handy tool that Katata and Yui discovered called pertussis toxin, and I think many of you know that you can use pertussis toxin to ribosylate and block the function of the GI-coupled receptors. So we extended our work on the muscarinic receptor to what then became a new, important, very interesting second messenger pathway, which was phosphoinositide hydrolysis. And when we looked at phosphoinositide hydrolysis in response to carbachol, in two systems we looked at, one was the heart and another was an astrocytoma cell, which I'll talk about later, we actually found out, along with our collaborator Ken Harden, that you could get carbachol to stimulate PI hydrolysis, but that response was not sensitive to pertussis toxin at all. So why does pertussis toxin not inhibit this PI response when the muscarinic receptor is meant to, apparently, from our earlier work, couple to and regulate GI signaling? So how did this make sense? Well, it turned out to make sense, as I think, again, many of you would know by now, that there are, in the first case, many flavors of muscarinic receptor. The one that we studied in the heart that couples to GI is the M2 receptor. There's also M1, 2, 3, 4, 5. Jürgen West and many others who cloned it discovered this. And the receptor that regulated phosphoinositide hydrolysis in the heart, in these astrocytoma cells at least, and it was a M3 muscarinic receptor. And how it regulated phospholipase was by coupling to yet another G protein, the third in the row, this one being GQ, pertussis toxin insensitive protein. And it was subsequently shown by John Exton, Paul Sternweiss, and many others that GQ actually bound to, regulated directly the activity of phospholipase, and therefore the formation of its important second messengers, calcium and diacylglycerol, should show here, which regulates phospholipase C. So being interested now in this sort of phosphoinositide response and having worked in the cardiac system, we wondered whether this phosphoinositide response might also be active in the heart. And it was. We could show that adrenergic and muscarinic receptors actually stimulated PI hydrolysis in cardiomyocytes. And why this didn't make a lot of sense is, for those of you who work in cardiovascular pharmacology, we know that there's a sympathetic and parasympathetic. They control contractile function and heart rate, and they do it by increasing cyclic AMP, calcium entry, SR calcium release, and then the parasympathetic limb inhibits that cyclic AMP pathway. But there wasn't any logical reason to think about there being a PI response which would go through IP3 sensitive calcium stores rather than SR calcium stores in the heart. So again, where did this lead us? How could we explain this? And this sort of led us into a whole area that became a major research area in my lab, which has to do with growth regulation by GPCRs. And it turns out that the agonists for GPCRs that couple to GQ, and there are many of them that work on cardiac myocytes, and among them are phenylephrine and ET1 and PGF2 alpha. Those agonists all act through GQ, and they all induce growth of cardiomyocytes, which is characterized by hypertrophic growth. The cardiomyocytes are postmitotic, they just grow in size. And these are actually to the same scale. This is the extent to which these neonatal rat ventricular myocytes grow, and you can see this organized myofilament that happens in response to the agonists, and then the green is a gene, ANF, atrial natriuretic factor, which is normally expressed early on, but is turned on, and as a number of fetal genes are in response to these agonists that act through GQ signaling. And to sort of further show that this really was something that GQ signaling was doing, we expressed through adenovirus GQ in the heart, so expression of the alpha subunit of GQ, which just like the agonists that stimulate it, induce this amazing hypertrophic growth of these cells, shown here again by the increase in size and the organization of the myofilaments there. We also, at slightly later, it was shown more directly by Nina Wedderschroek, Stefan Offermans, using GQ-GI knockout mice that they had generated, that hypertrophy induced by the sort of natural in a mouse model stimulation, which is pressure overload. Actually, the hypertrophy that you could get from pressure overload was ablated following conditional inactivation of Q11. So GQ11 signaling was clearly involved in pressure overload-induced hypertrophy, as it was in agonist-induced hypertrophy in the isolated myocytes. We did the sort of opposite gain-of-function experiment, where we expressed GQ in the heart in a transgenic mouse, and found that, in fact, the GQ did induce hypertrophy. But what it also did was to cause heart failure. And that was, at the time, sort of puzzling, and also has sort of led my work towards studying heart failure rather than hypertrophy. But the answer to this, well, let me just show, for those of you who don't know this, a little model here that, would sort of explain what the heart failure part of it, why is GQ signaling causing not just hypertrophy but heart failure, and that is, again, the question is right here, why is it causing hypertrophic growth, why is it causing heart failure as well as hypertrophic growth? And that's because that's what happens in real life. So in this model picture from an article written by Joe Hill, there's physiologic hypertrophy, which I haven't talked about, and pathologic hypertrophy, which is what I talked about, which happens with this pressure overload, or in the GQ transgenic with angiotensin with isoproternal. Pathologic hypertrophy, it's called pathologic, but it's actually also meant to be compensatory. The heart gets bigger and the walls get thicker and the tissue becomes better able to contract. But when this stress is sustained, this persistent stress leads to heart failure, which is characterized by thinning of the walls and dilation of the chamber, and an inability of the heart to contract normally, along with the development of arrhythmias. So that's what happens when it's sustained, and indeed, that's what we could mimic if we used, rather than the wild type form of GQ, a constitutively activated GQ. And there we saw these cells that don't look so happy, in which there was very clear DNA fragmentation. Here's the wild type GQ. Here's the constitutively active GQ, or the wild type along with an agonist, so you're getting agonist stimulated activation of the wild type, or here constitutive activation of the wild type. And that's what happens in vivo when this response mediated through GQ is sustained. This hypertrophic growth turns into heart failure. And in the talk I gave last night, I talked about the role of, let me just go back a second, of inflammation in the development of heart failure, but cell death, apoptosis, as is shown here, is also very clearly involved in heart failure. And I just showed you the DNA laddering. Here's some mitochondria that are very clearly disrupted. The whole structure of the heart is disrupted, in fact. Here's another picture, a pretty one that shows the sort of normal hypertrophic cell, and then some where become apoptotic, the cells burst, cytochrome C is released, and the cells become very funky. So that's what happens with sustained GQ stimulation in vivo, in vitro, and that's what leads to the development of heart failure. I mean, one of many things. So this sort of led to our general interest in, so this is this GQ system causing growth in myocytes, which, as I mentioned, they're postmitotic. This is just hypertrophic growth. They just get bigger. What about proliferation? What about other cells, like cancer cells? Does GQ signaling also lead to growth of GPCR, GQ signaling also lead to growth of other cells in particular? Other cell types. In particular, we were interested in cancer cells. And the cells that we'd been working with that I had told you about, these 132-1N1 astrocytoma cells, are actually human glioblastoma cells. So we'd been working all the time with the sort of cancer cell model. So we wanted to ask this question, and sort of happened that Silvio Gutgund, who I hired at UCSD and has now taken over as chair for me, published this paper that got us very excited, that said that the muscarinic receptor functioned as an agonist-dependent oncogene. And the receptor that he was looking at, we looked at all of them, the one that worked as an oncogene was the M3 receptor, so that was the GQ-coupled receptor that I said stimulates phosphoinositide hydrolysis. And so when he overexpressed the M3 receptor in, I believe he was using HEK cells or CHO cells, I can't remember what, 3T3 cells, when he overexpressed that muscarinic receptor, he got proliferation. So we thought that was really cool. Okay, we don't have to just study this cardiac myocyte, we can look at some cancer cells, and we went back to these 132-1N1 cells, these astrocytoma cells that we were studying, and we looked at the response to carbachol, which, as I mentioned before, is a very good activator of GQ and phospholipase stimulation and calcium and PKC and all of that. But carbachol was not a very good activator of cell proliferation based on here bromdeoxyuridine incorporation or here thymidine incorporation. On the other hand, thrombin was a great activator of proliferation in these cells based on these two readouts. And thrombin, as we now know from work by Sean Coughlin, Joan Trejo, who was in my lab at the time of this, works through a GPCR, the PAR1 receptor. So you can get GPCR stimulation of cell growth without overexpressing any receptors, just through this endogenous PAR1 receptor, but you don't get it really much through the M3 muscarinic receptor. Okay. And what is different about them? So it turns out, is that the one I was going to show next? So it turns out that thrombin induces Rho activation. And Rho is a small G protein. I got these slides in the wrong order, but it's a small G protein, and its activation can be measured by its GTP, by becoming GTP bound and using this sort of pull down assay. So thrombin activates Rho, and this proliferative response is actually dependent on Rho based on the effects of this C3, which I thought I was going to mention, but okay. So how is Rho I seem to be missing a slide or two, but it's okay. So how is Rho activated? Well, we had this notion that this newest member of the G protein family called G12, which was Mel Simon discovered many of these G proteins, and Mel up at Caltech had antibodies to G12, and we thought maybe G12 is activating the Rho pathway and causing proliferation in these cells. And this is before transfection or shRNA, but Mel had antibodies to G12, and Anna Aragay was working in his lab, and Jim Fermisco was in our department, knew how to do microinjection. So we microinjected antibody to G12 into the cells. We looked at thrombin-stimulated DNA synthesis. And here's the response to thrombin. And then here's the inhibition of that thrombin-mediated response with increasing doses of the G12 antibody. And antibodies to other G proteins didn't block this response. So it was clearly mediated through G12. Okay. And it then became clear, subsequent to that G12 was in fact involved, and how was, again, from something at UT Southwestern, a paper by Sternweiss and collaborators, in which they showed that G12 binds to and activates a Rho Jeff, whatever you like to call it, of which there were many, which is a GTP exchange factor for this small G protein, Rho A. So just like GS binds to cyclase, and GI binds to cyclase, and GQ binds to phospholipase, G12 binds to its effector, which is any of a number of Rho-GEFs, and then activation of Rho is the downstream response. So that's how Rho is activated in response to GPCR stimulation, in response to thrombin stimulation, for example. And then we turn to the question of how Rho signaling leads to cell proliferation. And Rho is initially described as a G protein involved in actin cytoskeletal rearrangement, and that indeed is something that it does in an acute way. But more chronically, Rho A elicits a number of transcriptional responses. And so we began to study this phenomenon that GPCR agonists that signal through Rho A elicit robust transcriptional responses. And our first clue that this actually happened, again in these cells that we were studying, was that we saw this very selective and sustained increase in this particular protein, CCN1, SIR61, at both the mRNA up here and the protein level. And it was rapid. Here, 3 hours, 6 hours, very robust. This is the, I believe, kind of the fold stimulation is something like, 50-fold. And we saw it with thrombin, which activates Rho, but with carbachol that doesn't activate Rho, and it was dependent on Rho. I'm not sure if I have that slide in here. And then this is the protein going up as well. And so we did a fair amount of work with this CCN1. I'm not going to talk about it, but basically it gave us a clue that led us a little further on, because when we talked about this protein, CCN1, or its homolog CCN2, called CTGF or SIR61, one of my colleagues in the department, Kunliang Guan, said, well, wait a minute, CCN1 is a canonical YAP-regulated gene. And so with Kunliang, did it first with LPA and showed that actually LPA, working through GPCR, activated YAP. YAP is actually not a transcription factor, but a transcriptional co-regulator. It binds to TEAD and turns on genes. And then we subsequently showed that with Kunliang that this also happens with thrombin. And then there's another transcriptional pathway that's also turned on through Rho A signaling, and Richard Treisman and Prius had did a lot of work on this. And this is this myocardin transcriptional coactivator, MRTFA. MRTFA is released from actin through Rho signaling. YAP, I should say, is dephosphorylated, but MRTFA is released from when G-actin changes, with the polymerization of G-actin changes, and MRTFA also enters the nucleus. It works through SRF, and both of those actually regulate expression of these CCN1 and CCN2 genes. So there's sort of canonical downstream genes turned on by Rho signaling through YAP and MRTFA. And we went on from there to show that these two transcriptional coactivators, YAP and MRTFA, are actually required for agonist-stimulated cell proliferation. Thrombin, S1P, stimulate proliferation, and these cells I've showed you here, 24, 48 hours, and here with YAP knockout or MRTFA knockout here by shRNA. I believe this is in the glioblastoma cells, but we went on from there to try to become more translational, which actually became a problem because my grant got transferred from GM to NCI, which was not a good thing, but we began to instead study G protein signaling in these patient-derived glioblastoma xenografts. So xenografts are made from glioblastoma cells, and the cells are grown up, and then you can put the cells back and look at tumor formation, which we did. So Olivia Yu is a graduate student, and Olga Chayam, who's now in my lab, is a professor from Brazil, trying to stay in the United States, who helped work on this project. And so the first paper that we published, we knocked down YAP and MRTFA in these stem cells derived from patient glioblastomas. The ones we used were called GSC23 PDX cells. We knocked down YAP, we knocked down MRTFA separately, two types of cells. We tried both of them, and they both were involved in the tumor formation, well, as shown here, and the proliferation. So here, just looking at sections of tumors at mitotic figures, you can see a mitotic figure blown up here in the control, and then much less cell proliferation in the tumors where YAP or MRTFA were deleted. And then we also looked, and this more compelling, at the tumors here by fluorescence here, and measuring the size of the tumor, which is shown here, the tumor fluorescence quantification. The tumor is much smaller when YAP or MRTFA are knocked out in these GSC cells that have been put back in the brain by orthotopic injection. So that's the tumors, that's the size, and then this is the survival, and then the blue is the control, and then the red and green are either YAP or MRTFA. The survival, the tumor's smaller, the survival's improved, the tumors grow less when YAP or MRTFA are knocked down. So from that point, we wondered what in vivo is activating these transcriptional pathways, logically GPCRs, and we looked at what GPCRs and G proteins are expressed in glioblastoma, and this is from TCGA, the Cancer Genome Atlas, and these pink lines are each one is a tumor cell, and all these things down here are GPCRs. I've just noted a few of them, and these are GPCRs that are known to couple to the G12 signaling pathway, the PAR1 receptors, LPA receptors, S1P receptors, prostaglandin receptors. All of these are overexpressed at the mRNA level in glioblastoma in the Cancer Genome Atlas. But what's most overexpressed, shown here, is the G protein itself, G alpha 12, GNA12, shown there. And actually, they're not overexpressed in the same, so the G12 is expressed in a number of tumors that don't have overexpression of the receptors. So the G12 is probably enough to itself elicit tumor formation or be associated with what happens in the tumor. And here's just, again, from the mRNA expression, non-tumor cells and tumor cells from glioblastoma with G12 mRNA expression, showing how much it is upregulated. And then the bottom figure here is actually showing that glioblastoma is in some ways unique, for sure, the highest in terms of G12 expression. So we sort of hit on the right tumor to study G12 expression. And then we reasoned that we would never know which of these receptors were in which tumors and which tumor cells. And so maybe logically, at least as proof of principle, though maybe less useful therapeutically, we would knock out G12 and see what happened. And so that's what we did. We deleted G12 in 2 lines of glioblastoma stem cells, the GSC23, which I've told you about, and another line which we just studied as a second line, the HK281 cells. And we were sort of dismayed that when we put these GSC23 cells in which G12 was deleted into the brain, we didn't see any difference in survival. And then these are two separate experiments where we looked at tumor size at different times, again, through measuring the fluorescence, and the tumor size didn't change either. So a little bit different than what happened with the YAP or MRTFA knockdown. But we reasoned, because you have to reason, that probably there were many other factors that could stimulate activation of YAP or MRTFA. But what we did see that was very striking is that these orthotopic xenograft tumors that had G12 deletion showed very marked decreases in invasiveness. So this is the border here of the tumor cell. It's an H&E stain. Here's another one, border of the tumor cell. And then this is the border of the knockdown, and another, the border of the knockdown. So you can see that the borders are much smoother. There's far less invasiveness when G12 is deleted. And then this is shown in another way. This is using an antibody for human cell nuclei. So these dark stained cells in this immunohistochemistry down here are just the GSC cells, which are human. The other cells are mouse cells. And so again, you see this smooth tumor border when G12 is knocked down, and a much more invasive tumor border in the control. And why this is particularly important for this tumor is that it is the invasion of the tumor cells into the stroma, into the border, that makes it very hard to remove this tumor, glioblastoma, which is the most lethal of any brain tumors, and it's very hard to excise it because it has invaded the stroma, and you never can get the size out, and so there is recurrence of the tumor. We also found that in these tumors, there were changes in two types of genes that we looked at, cancer stem cell genes here, and a number of cancer stem cell genes, OCT4, PAC, SEX2, it doesn't matter to name them, but a number of these stem cell genes showed diminished expression in the G12 knockdown, shown here in the blue. And also EMT or PMT, so PMT is proneural mesenchymal transition, the equivalent of what happens in other solid tumors called epithelial mesenchymal transition. And many of these genes also changed in the direction of them being more proneural, which means sort of less mesenchymal, which means less invasive. And so both these changes in these stem cell genes, as well as changes in these genes that make them more or less aggressive, are likely causes of the diminished invasion. But rather than looking at any of these genes, we did a more global search using RNA-seq to see what other genes might be regulated in the G12 tumors. And this is the in the volcano plot, there's a heat map here. You can see that there's 500 or so genes that are actually up or down regulated. These are the down regulated genes. This is the G12 here, which should be down regulated. And then this is just one that we picked out called thrombospondin. And thrombospondin was interesting because it was so highly down regulated in the absence of G12, and we had discovered that gene before. It's a gene that's mostly been implicated in angiogenesis, but it also plays a role in invasion. And we showed in data I'm not going to show you that G12 activation also in cells induces expression of thrombospondin. But I think, and this is my almost last slide, that interestingly, thrombospondin is also very high in glioblastoma. Shown here in the middle, it is associated, correlated with the expression of G12, particularly in these mesenchymal, highly invasive tumors shown here. and shown here below. And so we as sort of proof of principle, knocked down thrombospondin in the GSC23 cells, put them back into the mouse, looked at the tumor. Like what we saw with G12, the mouse survival tumor size didn't seem very different, but we again saw that the border was very smooth rather than invasive, as in the control. So I've walked you through my life in G protein signaling from point A to point D, and kind of pointed out how we learned about the role of these receptors and these G proteins in cardiac growth and death and heart failure, and in cancer cell proliferation and invasion. And probably as my first major take-home lesson, you can enjoy an exciting 50-year career if you notice, as Alice did, and this is a quote, that what could be seen from the old room was quite common and uninteresting, but that all the rest was as different as possible. And it was these differences that kind of led us through the path that I described to you to discover at least a couple of important things: that GPCR activation of GQ is a prominent player in the development of cardiac hypertrophy, inflammation, which I didn't talk about here, and heart failure. And then the GPCR activation of G12 is a critical mediator of transcriptional responses in cancer cell growth and invasion, the targeting of which has, I think, therapeutic potential, if we could figure out how to do it. So just to particularly acknowledge a couple people, I mentioned Olga and a longtime collaborator, Shigeki, ongoing support from NIH, and thank you for your attention.
Thank you. That was absolutely fantastic. A very worthy keynote. We're now open for questions. Please, everyone, there are microphones just at the edges of the lecture hall, so please go up to those microphones to ask your questions. Perhaps I'll kick off. I was just wondering, in your translational experiments where you looked at survival, where you had deleted silenced MRTF. Can you not hear? So in your survival experiments, the translational work, where you had silenced MRTFA or YAP, the survival was improved, but actually not completely. And I just wondered, what other pathways you think were being activated? It just seems that there's, well, in the tumors, but it sort of relates a bit to cardiac. You have GQ, GS, G12, and when you take out one pathway, is there upregulation of other pathways to compensate? And I'm particularly thinking about heart failure, in fact, where in patients we know that endothelin levels go up, yet endothelin antagonists have not translated well. And is it because there are other G protein pathways that are being upregulated? Is it true of the glioblastoma, just as it is of cardiac hypertrophy?
Sure, yeah, I mean, I think that's clearly right. There's probably upregulation of other pathways. And then there's sort of this phenomenon of you need so many things, so many alternative pathways to make sure that that something else will take over if you knock out something, or you need to knock out, if you knock out A, you'll knock out everything, but you also need to knock out, if you knock out B, you'll knock out everything. So there's just a lot of redundancy and compensation, as I think you're suggesting. Yeah.
Yeah, I mean, John McMurray actually in his talk, he basically said that we need to be targeting lots of different pathways at the same time, perhaps instead of bringing in one drug at a time, bring in several drugs.
Right.
Maybe it's true of both scenarios.
Sure. I mean, and I guess that's the only reason I'm thinking that, if we were targeting a receptor in the glioblastoma, we wouldn't know which one to target. If we could target G12, which arguably hard to do, maybe, CRISPR gene editing or antisense or something, because you could get into that tumor, then you would potentially, if you believed that the G12 part was important, block any of those receptor signaling pathways. I mean, we can't really do that in heart failure. But in a tumor, setting, you potentially could. So.
There's a question there.
Hi, Joan. It's Graham.
Hi, Graham.
Where are we with actually trying to get regulators for G12? Because clearly the development of the FR compounds and the YM compounds have been remarkably interesting for targeting GQ and G11. Clearly people are working this. Could you give us a little bit of an update of your understanding?
You know what? I don't actually know. The only thing I've thought about and talked to people about at what used to be called ISIS, now Ionis, is doing antisense because they've done a lot of that. But I honestly don't know if anyone has looked at or worked on drugs to target G12. So let's do it.
Well,
yeah.
Have a word with Evie Costinas next time you see her.
Okay.
Please.
Hi, Joan.
Hi, Andrew.
A great talk. I guess my question isn't really a question. It's just to get your thoughts. You described very beautifully, particularly at the beginning, how you did experiments which you then expected to see a particular result, and with the M2 receptor, for example, in the heart that you showed, and you didn't get it. Okay, so and then you've got something actually completely the opposite. So I just wanted to ask you about the way that you thought about those experiments, whether you thought you were just wrong, whether you thought you were right and everybody else was wrong. How did that work out in the way that you just painted that beautiful picture of your career?
Right. So I remember, and then only a few of you will be old enough to have done your cyclase assay with a scintillation counter, maybe. So I remember looking at my counts, my quick counts, because we'd run them through real fast so we could see the data, and thinking, no, I must have mixed these up. It can't be the dopamine is stimulating this and norepinephrine is not. So there I thought I was clearly wrong. And the cholinergic inhibition, I initially had not seen it with acetylcholine. I'd initially actually seen it in some bizarre way that I was studying depolarization. And depolarization turned out to be releasing acetylcholine. And then, we could show that it happened with carbachol. But I just thought it was something else with depolarization. And it never occurred to me that it was acetylcholine. I thought depolarization was what was, maybe inhibiting the cyclase in some bizarre kind of way. The pertussis toxin failure on the PI response. Well, I was working with Ken Harden. He's a really smart guy, and I think we were kind of cool enough to think maybe at the same time these other muscarinic receptors were coming around, and maybe it was a different pathway in a different G protein. And I think we actually had shown that there was sort of G protein stimulation of phospholipase in broken cells. Fain had shown that, and others had. So we kind of thought there might be another G protein. So in some ways, for sure. Certainly in my youth of youth, I thought I was wrong. But then I guess I just thought, as I wanted to say in my last talk, hey, this is interesting, and the lesson I kind of want to say is, as follow your unexpected results, because they're the ones that turn out to take you further. Thank you for that question. Thank you for the opportunity to answer that question.
And I just wanted to ask, everyone's becoming much more aware of the need to explore pathways in both sexes. And particularly with cardiac hypertrophy and heart failure, sex differences in susceptibility outcome with heart failure now is very much talked about. In terms of the G protein pathways, is there a preponderance of one pathway over another between the sexes? Do you know? Is there any evidence that...
Guilty as charged. No, I write in my grants that I'll, look at sex differences if there are some. I usually, use males. I'll sometimes, throw in both, and if they're not different, then I can sort of justify that I'm just using one or the other. I'm sure there are differences, and I'm not the one who studies them, but in terms of the cardiac field, as you know, there are vast differences. Whether they translate into differences in G protein signaling or expression is, a good question, but have no answer.
Okay. Well, if there are no further questions, it just leaves me to thank you once again for an absolutely fabulous lecture.
Thank you for having me.